everyone. In this video, we're going to describe some important components of the cell life cycle. And as we begin, I thought we would just sort of talk a little bit more about the nucleus, because the nucleus is the the most important component when we talk about cell replication. So I want to highlight the recognizable features of the nucleus. And in this drawing, we can see the nuclear envelope, which is basically a double membrane that is separated by fluid. So it's basically two-layer membrane, and the outer portion of the membrane is connected or continuous with the rough endoplasmic reticulum. So later when we talk a little bit about protein synthesis, we'll see the significance of this connection. And the inner membrane is actually lined with these particular proteins called lamins. So it's called the nuclear lamina, and lamins are basically the structural components that make up intermediate filaments. So we know that intermediate filaments is one of the parts of the cytoskeleton, and this is just a specific type of protein we use. The second feature is a nucleoli. So it would be nucleolus, singular, nucleoli, plural, and there's usually one or two per nucleus. One important feature of the nucleoli is that it's the site where ribosomal subunits are created or synthesized. We also notice that there are nuclear pores, which are basically aqueous transport channels or gaps between the nuclear envelope that allow for movement of materials into and out of the nucleus. So mRNA is going to be moving out from the nucleus into the cytosol, and larger molecules and other proteins will be coming in. And of course, because it's just an aqueous transport channel, we have free exchange of water and other ions. The next feature is chromatin. So chromatin is about 30% DNA. It's about 60% histones, which are proteins that I'm going to draw in a second, and about 10% RNA, which basically we can talk a little more about when we talk about protein synthesis as well, but will be new or newly forming. So what I'm drawing next is a example of histones. Histones are almost like little round discs. So this is 4 of them all together. And we know that DNA normally is in the alpha helical arrangement, but actually when we there's just so much DNA, it's so long that we coil it very, very tightly, and we actually pack it around the histones. So 8 histones, there is sort of like 4 and then 4 we can wrap twice around, and that actually is called a nucleosome. And so nucleosome, 8 histones plus the DNA. And think of it kind of like DNA is almost like, if you think of Christmas lights, and Christmas lights being unbelievably tangled if you don't sort of sort them and pack them in nicely at the end of each holiday season, it's very difficult to sort of access them, and then you spend a lot of time untangling the next year, and it's a disaster. That's how DNA would be. We need DNA to be usable, so it's not always the most usable if it's just in a big jumble. So when we want to pack it down nicely, we wrap it around these histones, and there's other sort of coiling methods as well. And then when we need to use it, we can sort of unwind it so we can access the individual DNA fragments. And the last feature of the nucleus is the nucleoplasm, which is just basically the cytoplasm of the nucleus. So I want to remind everyone that we are talking about somatic cells as opposed to sex cells. And somatic cells contain 46 chromosomes. And the 46 chromosomes are really 23 pairs of chromosomes. So they'd be numbered 1 through 23. 23 from mom, 23 from dad makes 46 total. So the chromosomes are basically a tight packing arrangement of the DNA. So the DNA is organized into 46 chromosomes. So when we think about cell replication, we really want to make sure that we can copy the DNA. And we need to copy it exactly, because if we make mistakes copying the DNA, then it's possible that something won't work right in the new cell. So the DNA copying is very critical. When we make a copy, we can actually see that there's sort of 2 clumps or strands, and those are called chromatids, and they're connected at the center by a centromere. And we'll see the significance of the centromere shortly when we consider the actual division, the actual cell division process. So one critical point about DNA replication is that it actually follows a semi-conservative replication model. And I just want to sort of talk a little bit about that for a second. So basically, if we sort of draw the DNA a little bit stretched out, when we want to copy it, we can completely unwind it so we can have easy access to both strands of the DNA. And having access to the strands of the DNA allows us to copy them. When we're doing cell replication, we're going to copy all of it. And later we'll show that if we want to make a protein, we'll just make a copy of some of it, and we'll sort of use it for later. So in this drawing, we can see that we have unwound the DNA, and we can see that it's almost like if you think of undoing a zipper, it's been unzipped, and we have these two sides with like the teeth on either side. And what we're going to do is we're going to bring in new nucleotides and string them along and make sort of the opposite of what was already there. So the red on one side matches the purple on the other side, and also vice versa. And so we actually are getting two new sets of DNA, but each new set of DNA has one old strand and one new strand. And this is semi-conservative replication. We are basically retaining one half of the DNA for each new piece, as opposed to just sort of making a copy and one is old and one is new. And this reduces the chance of making some mistakes. So when we look back up at our chromatids, when we fold all of this new DNA up, one set makes one side, or one chromatid, the other set makes the other chromatid, and then we have, again, joining at the centromere. So now let's talk a little bit about the cell life cycle. So I think it's easy to understand that cells, just like humans, will grow and eventually they can get old and they might die, but or if there's an opportunity, we might be able to sort of recycle some of the parts and make some new cells. So the cell cycle is basically divided into G1, S, G2, and M. And these are all just phases, and we'll just talk a little bit about each one. And we can see that as sort of a spin-off. Maybe instead of going to G1, we can go to G0. So G1, it actually the G stands for gap. It's a gap phase. Gap phase 1 is basically normal cell functions and lots of cell growth, and also replication of centrioles. So we mentioned before that not all cells have centrioles, and centrioles are critical for cell replication. And so even in the early stages of preparation, we will have centrioles present for replication. S phase is, it actually stands for synthetic, and this is the synthesis of new DNA, basically. So we're making our copy of DNA and preserving the integrity of the DNA as much as we possibly can. This is followed by G2 phase, also G for gap, which is associated with further protein synthesis and some final preparations, such as the centrioles kind of being ready to go. So I want to just sort of highlight quickly the something called the centrosome. So we have our cell, and then we have our nucleus, and nearby the nucleus is a centrosome, and it contains two centrioles. So two centrioles per cell. And then when we talk about in G1 replication of the centrioles, we're basically going to make a second set, so a second centrosome. So there'll be 4 centrioles total. And this is important because the centrioles are the site of microtubule assembly. And when we talk about mitosis, which is the M phase, we're going to need to make some more microtubules. We're going to just show that in a second. There are 4 phases of mitosis. So mitosis technically refers to division of the nucleus, but it's this is the actual part that's cell division. So all the other parts are just sort of cell growth and function, and we get to mitosis, we have cell division. So so the first phase is prophase, and during prophase we're going to form something called a mitotic spindle. And so basically at those centrioles we're going to assemble some microtubules, and we're going to make this structure. And also, so in this cell, we're seeing just one chromosome replicated, but you can see that we have 2 chromatids, and we have they're connected at the centromere. And the nuclear envelopes, remember the nuclear membrane, during prophase is going to disappear or dissolve. And this will allow the mitotic spindle that's forming to have access to the chromosomes, because they're going to help to sort of organize those chromosomes into 2 new cells. So prophase is characterized by early prophase and late prophase, and as we move to the later stage of prophase, we can see that what's going to happen is the individual microtubules of the mitotic spindle are sort of reaching out and trying to find the chromosome. So they're trying to associate with the chromosomes, and what they're specifically looking for is that centromere. So when it's able to contact the centromere, that microtubule sort of hooks onto it and it says, okay, like I'm ready, I've got it. And so all 23 pairs of chromosomes are going to sort of be connected to the mitotic spindle. That's the goal. It's going to take a bit of time, but once we get it situated, we'll be able to sort of move on to the later stages of mitosis. The second phase is metaphase, and metaphase is characterized by lining up the chromosomes along the midline or the metaphase plate. So basically, we've organized all the chromosomes that we've captured now, all these duplicates, right along the midline, so it'll be easy to sort of see that we're going to split the cell in half the DNA, and then later half the cytoplasm. Metaphase is followed by anaphase. Anaphase is characterized by splitting the chromosomes at the centromere, so the individual chromatids are being pulled toward the two opposite poles of the mitotic spindle. So now we're kind of dividing them into different ends of the cell so that we can prepare for final separation. And then lastly, we have telophase. And telophase is characterized by the reforming of the nuclear envelope. So we can see that the DNA is going to be separated into the two sides and then contained within a nucleus. And sort of like partway through anaphase and then also through telophase, we have another process called cytokinesis. And cytokinesis is basically the division of the cytoplasm. And so what we will get is, I'm going to draw in a dotted line, and that represents a cleavage furrow. And so basically it just sort of, it's almost like the two parts are closing in, and then eventually they're just going to completely make two different cells and it'll split apart. So we can see at the very end we have our individual two new cells each with their own nucleus and a full array of DNA that is needed. I wanted to mention before about G0. So some cells might opt out of the cell cycle where they have this sort of continuous growth and then division and then growth and then division thing happening. Cells that are in G0 phase are really, they're just not going to divide, but that doesn't mean that they can't divide later. And so a good example would be liver cells. The liver cells are not actively dividing, but if you were to cut out part of the liver, then the liver will grow back to its normal size because the cells will enter into the cell cycle. Some factors that will trigger cell division include an increase in surface area to cell volume ratio. So if you think about the fact that inside of the cell there'll be a number of organelles that need to receive oxygen, glucose, nutrients, and things from the outside of the cell. And if you think about the mechanisms that are available to bring that stuff in, the larger the volume of the cell, the less the surface area or the area of the membrane has the ability to accommodate the volume of needs for the cell. So as the cell gets too big, there's just not enough entry points for everything. And so, and the diameter is too big, so diffusion inside the cell is poor. And so the cell just isn't able to get all the nutrients it needs. So that could be a trigger for cell division. To make two smaller cells, it would be increasing the amount of cell membrane relative to the volume, thus making that part easier. Another example would be presence of certain chemicals, such as growth factors or hormones can trigger cell division. And lastly, something called contact inhibition. So the cell knows that as it sort of expands, that there are cells sort of nearby, but when it gets to a point where that cell is starting to touch all of the cells near it, it's starting to feel a little bit like there's just not enough room for it anymore. And again, the cell will divide because then we're making two smaller cells. And so contact inhibition sort of inhibits further growth before the next cell division by triggering cell division a little bit more frequently. So I think we could agree that the cell cycle is normal And it occurs all the time, even in the absence of stress. But when stress comes into play, the cell might have to make some accommodations to deal with stress. So stress is defined as some sort of strain or damage in response to a stimulus. And adaptation is basically structural and functional changes to the cell in response to stress. So I want to just highlight a couple of examples of adaptation because I think that they're very relevant, and they often have physiologic and pathologic associations with them. So the first one is hyperplasia, which is an increase in the number of cells in a particular tissue or in a particular area. Triggers for this could be hormonal, so in response to certain hormones. So for example, during pregnancy, enlargement of breast tissue to accommodate the impending expectation of breastfeeding. Also, another example would be compensatory. So this would be in response to some sort of injury or loss of tissue. So earlier when I mentioned if you have like part of your liver removed, then compensatory hyperplasia would be increasing the number of cells, increasing that division, entering back into the cycle, etc., so that we can make up for the loss. The second type of adaptation is hypertrophy, and hypertrophy is an increase in the size of the cells. This could be physiologic or pathologic. And so an example of physiologic would be an increase in muscle mass, increase in the size of muscle cells in response to exercise. And something that would be pathologic would be something like left ventricular hypertrophy or hypertrophy of the heart. So if you have chronically elevated blood pressure and the heart is working very hard to pump against it on a regular basis, then the cardiac cells are going to grow in size to sort of accommodate that. And that's in response to sort of a disease process. So that's why it's pathologic. Next form of adaptation is almost the opposite. It's atrophy. It's a decrease in the size of a cell. And it could be due to a decrease in the workload of the cell. It could be due to a decrease in blood supply to the cell, decrease in nutrition availability, decrease in certain hormones. It also could be loss of neural control. So like later we'll see, the importance of neurons and the nervous system controlling muscle contraction. So if we don't have neural control, then we don't, maybe use the cells that we want to use. And also aging would be another example that could lead to atrophy. The next would be metaplasia, which would be a change of one cell type to another. And this could offer some sort of protection. So if one cell type is maybe a little too delicate, the body will sort of transition to a different cell type to be more protective. And then lastly, we have dysplasia, which is basically abnormal or disordered cell growth. And this often is a precursor to cancer. So cancer cells are very, they're sort of dominant. They're really, I guess maybe we could even say strong. They steal resources from the body and they proliferate very quickly. Now, also when we think of cell adaptation in the cell cycle, we need to think of cell death. So normal cell death, which is sort of an expectation from time to time, is called apoptosis. And it's basically cell suicide. It's a programmed death. And it's actually really interesting that the way that it's controlled, it's sort of just like almost like self-digests, but it doesn't leak any of its contents into the external environment, so it doesn't affect any other cells around it. So it really just sort of shrinks away by itself. But when we think of accidental cell death, or we think of some sort of stressor that adaptation fails and we get to death, we actually call that necrosis. So this is more like cell murder. And some examples of triggers for necrosis would include hypothermia, hypoxia, decrease in pH, or other cellular trauma. And in this case, we get typically swelling and then cellular lysis. So this is going to burst and expose all of the contents to the other cells. So much messier in terms of a death, but it is an accidental death, as we mentioned. Okay, so that concludes our explanation of what happens in the cell cycle. And coming up next is going to be some information about protein synthesis. So I hope you enjoyed it. Thanks for watching.